Good afternoon to all. I respectfully thank our most respected chairman for giving this opportunity. Thank you, sir. It is my absolute pleasure to introduce our esteemed guest, Dr. S. Juby, Associate Professor in the Department of Pharmaceutical Chemistry at JSS College of Pharmacy, Ooty. Dr. Juby is a renowned educator, researcher and innovator with over 22 years of experience in teaching and research. Dr. Juby completed her B.Pharm from Sankaralingam Bhuvaneshwari College of Pharmacy, Sivakasi, and her M.Pharm from KM College of Madurai, where she was recognized as the best outgoing student. She further pursued her PhD from JSS Academy of Higher Education and Research, Mysore, solidifying her expertise in pharmaceutical chemistry. Her research focuses on discovering novel chemical entities for cancer therapy using advanced in silico tools and drug repurposing, making significant contributions to the pharmaceutical field. With 88 international and 34 national publications and an impressive cumulative impact factor of 155, Dr. Juby's work has gained wide recognition, accumulating over 2,071 citations and high index of 22 on Google Scholar. She holds 7 granted patents, has filed and 7 more. She has authored 5 books and 8 book chapters. Currently, Dr. Juby is leading 3 funded research projects sponsored by AICTE, ICMR and JSS Academy of Higher Education and Research. She has also successfully managed several other grants, including one international grant from Sakura University, Saudi Arabia. Dr. Juby is a committed mentor, having guided numerous PhD, M.Pharm and B.Pharm students. She actively involved in institution and accreditation activities for other organizations like NAC, NBA, NIRF, showcasing her leadership and dedication to academic excellence. Her professional accolades include prestigious awards such as Pharmacy Woman of the Year of 2023, Young Researcher Award 2019, Best Teacher Award 2022. Beyond her academic achievements, she is life member of prominent organizations such as IPA, APTA, SBTA, contributing to the advancement of the pharmaceutical profession. Dr. Juby's philosophy is deeply inspired by Buddha's words, "The mind is everything, what you think, you become." Today, we are fortunate to have her share her vast knowledge and experience with us. Please join me in welcoming extraordinary Dr. S. Juby. Welcome, ma'am.
Thank you, ma'am.
A very warm good afternoon to one and all present here. So at the outset, I thank the chairman sir, Ultra College of Pharmacy, the principal, dean, vice principal sir, and eminent speakers and my respected seniors, Kavimani sir, Sambat sir. And so I'll be taking you to the next 45 minutes to 1 hour. One of the AI tools, that is the molecular modeling tools, how you can integrate the molecular modeling tools to your pharmaceutical research. I think for the past 1 hour, sir has enlightened the enormous AI tools. Okay, so how you can effectively take this AI tools for your field, especially pharmaceutical field. Okay. So one of the AI field is the computer aided drug design or various molecular modeling tools. So before going to my talk, I will be telling a fact that normally the students know you have one fact in your mind that we have to spend like exclusively, or we have to spend money for buying these tools, or we have to get, not like that. These are all easily available online tools. Okay, so how you people, you are very good in chit chatting with your mobile phones. So with using that mobile phones, how you can take effectively your research. So that's what I am going to do it. Okay, so and sir rightly mentioned the various tools, no? So you can operate any tools, but you should have some basic concepts, basic principles. So without that basic principles or basic knowledge, no, if you get the results also, it won't be beneficial. Okay, so you should know the concepts and principle. So first half an hour for some important basic tools I will be taking you through. Then the next 15 minutes I will be giving a short case study. Okay, it will be interested, so you can also think it of how effectively you can use these tools. Okay, yeah. So if you have any doubts, you can stop me at any point of time and I am happy to answer it. So before that, I just wanted to know how many of you are PG students. Please raise your hands. Just so this is the roadmap of my presentation. So I will be giving an overview of CAD, computer aided drug design, and as I told you, some basic tools which you can available on, you can use it, you make it use through online. Molecular docking, in silico ADMET predictions, pharmacophore modeling, visual screening, molecular dynamics, and I will be ending with a case study. Yeah. So before coming this AI CAD, so we were completely depending upon the traditional drug discovery. Okay, I think you might be knowing the very good example of penicillin's discovery, correct? So he never knows that he will discover penicillin. It is an accidental drug discovery. Like that, so we have a traditional drug discovery system, okay? Without knowing whether the compound will give the activity or whether this isolated compound will give it can be used for this disease, people used to synthesize lot of compounds, or people used to isolate lot of compounds, okay? Then they will go for in vitro testing, then they will go for preclinical modeling, and it will come to clinical trials and all the phases. So this is the traditional drug discovery. You can see here the lead generation, the biological testing. If promising, it will be going for the next phase like clinical trials. Okay. Otherwise, again the drug design cycle, it will be going on. So now this traditional drug discovery is supplemented by the word computer aided drug design, that is the rational drug design. So what is this rational drug design? So this is all completely based on the molecular targets. Okay, so we should have a knowledge about the target. The target is nothing but it is a biomacromolecule which is involved in the particular disease. Okay, so it may be a protein, it may be a receptor, or it may be a ion channel. Okay, so this rational drug discovery is completely depending upon the target, molecular targets, which is assumed to be participate in some disorder. Okay, so suppose if I want to find a new molecule against, okay, against HIV virus, okay, so I should first completely study the pathophysiology of the disease, the molecular pathway of the disease, and I may directly get into some target which is the most like very much relating to that particular disease. It may be HIV protease, okay, or reverse transcriptase. So all you have to be completely thorough about this target. Okay, so till 1980 people have used computers only for data handling or data storage. Slowly people have used the computers in the pharmaceutical research. That's what the area is called as computer aided drug design. So this CAD is a combination of various disciplines. Okay, so it is a combination of IT, information technology. I think you people are well versed using these computers, mobile phones, all those things. Of course, you should have knowledge about physics, because all these things are drug receptor interactions. Then chemistry you should have, biology, medicine, okay. Then, yeah, I am very good in maths. You can do QSAR studies, okay. You have so many available online tools for 2D QSAR and 3D QSAR, okay. So it is a combination of various disciplines, okay. And the picture is a representation, what is. So all these things, these CAD techniques are simulation. How the receptor, it is, how the drug, it is interacting into the receptor in our body, so that you will be doing in front of sitting system. All it is simulations, okay. So that's all a little bit introduction about CAD, and this is the process I think you all might be knowing. So first of all, you have to identify the disease protein. As I told earlier, the target. Okay, selection of the target is very, very important. Then you have to determine the structure of the particular target. Okay, so you have so many tools available, and we have the data. Okay, only thing is how we are retrieving it from the available databases. Okay, then identify active site. That is very, very important. Okay, then you can go for virtual screening of compound libraries. Okay, so usually screening means what? A testing of molecules, correct? Testing of molecules against for a particular disease. Okay, so virtual screening. Okay, so labs together molecules are available. We have so many databases. You can take the data compounds from the database and you can do the virtual screening. Okay, and then you can go for synthesis of lead compounds. Okay. No ma'am, I am not very good in synthesis. Don't worry, you can purchase the compounds. So many compounds are available. You can purchase the compounds, and some people are really wanted to get it from the natural source. You can go ahead with those compounds. Okay? Synthesis or isolation, optimization, then clinical trials, and at last it will come as a drug. Now, after this CAD come into this picture, the money also has been reduced, and the years, okay, the drug discovery years, almost it will take 15 years, 10 to 15 years, and the money involved is around 2.5 to 3 billion dollars. Okay, so initial period, the lead generation period, so that period has been reduced once this CAD has come into the picture. So these are all some advantages: cost savings, time to market, better insight. Okay, why? Because you are completely analyzing the disease and you are completely analyzing the target, then only you are discovering or you are designing the drug. Okay, so you are playing around with the target. Based upon the requirement of the target, you are finding out some molecules. So you will be having always better insights. So these are all some examples. Okay, you can see here from 1981 the CAD has started. Okay, so starting with captopril, okay, ritonavir, Okay, within 7 to 8 years, the FDA has approved, okay, the antiretroviral drug. So the list goes on, okay, the various drugs discovered through this CAD. Okay, you have tenofovir, gold, so goldiracin. So all these drugs have been discovered through this CAD. Yeah. Now, these are the two areas. Okay, the drug design you can broadly divide into structure-based design and ligand-based design. Okay, so the structure-based design, okay, you know the complete details about the target protein. Okay, I will be explaining or I will be using these two words again and again, target and ligand. Okay, so based on the requirements of the target, you will be designing some molecules, that is structure-based drug design. The another one area is ligand-based drug design. Okay, you have so many compounds in your hand. Okay, or you have so many hypothetical molecules in your hand. Okay, you can very well go ahead with ligand-based drug design. Okay. Yeah, this is a slide which shows all the molecular modeling tools. Okay, so you have two probabilities. If I have the structure of the protein, okay, so you can go ahead with structure-based drug design. So the structure-based drug design can be subcategorized into ligand docking. I think most of you heard about the word molecular docking. Is that right? Have you heard? Yeah. Then de novo design. Have you heard that word, de novo design? Fragment-based drug design. Okay, you have bits and pieces of so many fragments or scaffolds. With that, you can only build your ligand. So that is called as de novo design. And you can do MD, molecular dynamics, and pharmacophore modeling, where today I am going to take you to the pharmacophore modeling. Okay. And the right hand side you have ligand-based drug design. Okay, so where you don't have the information about your target protein. In that case, you can go ahead with QSAR, quantitative structure activity relationship, pharmacophore modeling. It's an excellent tool. You have ligand-based pharmacophore modeling and structure-based pharmacophore modeling. Then you can go ahead with ligand-based visual screening. Okay, so any side if you do it, you will be ending up with propose new existing or you will be optimizing a new lead molecules for the for any particular disease. Okay, so at the end of your CAD, you will be having hits. You will be having hits in your hand. With that hits only, you will be going for either synthesis or you will be isolating it, or directly the hits are available in the market. You can buy it and you can go for activity checking. Yeah. So most of the time we will get doubts or we will get confusions. Which drug design we should select? Whether structure based or de novo design or ligand based. Okay, so these are the various scenarios. If you know the receptor structure and ligand structure, you can go ahead very well with structure based drug design. Okay, if you know the receptor but I don't have any idea about the ligands, you can very well go ahead with de novo drug design or fragment based drug design. Okay, the third scenario is I don't know about anything about the protein, only I know the details about the ligands. Yeah, you can go ahead with ligand based drug design. So these things only we always get some confusions, or which of course each and every one are excellent in any one of these, design. So you can choose any of these. So when all it all started from 1970s. Okay, the people, they are very confident about the three-dimensional structure of the protein. Okay, they have crystallized the protein. They have isolated from various sources. They have crystallized the structure and they have deposited those structure in some databases. A very good example is protein data bank. I think you all be aware of that. Okay, you can see there n number of structures, three-dimensional structures of the proteins are fed into that protein data bank. So then only all these structure-based drug design, it started. Okay. So this is molecular docking, a very basic tool in your structure-based drug design. Okay, so what is molecular docking? It is a definition. Of course, I am not going to read it out. It is just an operation between two molecules. Okay, one will be the target, a macromolecule, and another will be a ligand. It may be a small molecule. So how these two molecules are, what I can say, binding together. That's all about this molecular docking. And how much the binding, you are going to quantify it as well as you are going to study qualitatively also. That's what the molecular docking. Okay, so you imagine you have a big protein, okay, and you have a small ligand. So how these two are interacting with each other? Okay, biologically, how it is happening? Okay, so that your docking tool, it will give you the simulation. Okay. So here you are finding out two things. One is the binding. You are going to quantify the binding affinity and you are going to study the interactions. Again, interactions means the protein is made up of so many amino acids. Each protein has got some active site. That active site has got some residues. Okay, so which residue is very much essential for the activity. Okay, so that's what about this molecular docking. Yeah, very simple. Always I give this example to my students. You also might be knowing it is a very basic lock and key mechanism where the lock will be your target, your protein, and key will be your ligand. We know very well all the keys are not, it won't open all the locks. So, and the second thing is, you have to keep the key in proper direction. That's what we tell the conformation. It should be in the proper conformation, then only it will open up the lock. It's a very basic theory, lock and key theory. Of course, afterwards, no, it has so many theories are there. Induced fit theory, quotient theory, then conformational selection theory and all. So, the very basic understanding is this lock and key theory. Yeah, this is very important. You got your protein, you got your ligand, okay, and you have your tool also, your molecular docking tool also. So what are the things it has to come to your mind? So first thing you have to see, keep a compound in approximate area where binding occurs. And what are all the things you have to evaluate? Do the molecules bind to each other? That's a very basic question. Okay, whether these two molecules are okay, they are binding together. That's what the first thing. Okay, the next one is what? If yes, yeah, you have that there is binding occurring, then what you have to do it? How strong is the binding? That's what all these scoring, dot score, all these poses, all these interactions, all these things are coming there. How strong the binding? whether stronger binding affinity, again the stronger, weaker, all these things are quantified by some numbers, mathematical numbers. Okay, so you have some mathematical values for each and everything. And one more thing I wanted to tell here, you have to validate each and every method what you are doing it. Okay, of course you have some validations also. So the third thing is, how does the molecule or protein complex look like? Okay, it is all understanding the intermolecular interactions, and last you have to quantify the extent of binding. So these are the process. And these are some theories. So as I told you, two things. The first thing is the process or the two-dimensional interactions. You have to do it. So it is a blend of these two, sampling and ranking. Okay. So ranking, again, it will give you different scores. So based on the scores, you have to rank the molecules. Okay, it is the blend of these two. So the docking, it can be between protein and ligand, protein and protein, and protein and nucleotide. Okay, so the first one is very much obvious, and people will go for this protein and ligand, the molecular docking. This is a typical docking workflow. You go with any tool. It may be commercially available or freely available. This is a typical workflow. You have to select the target. Okay, then you have to prepare the target. That is very, very important. This particular software, it will take you. Okay, it will give you some guidelines. Very basic example I will tell you. When the scientists, I told you the protein structure, how the people have got, they might have crystallized. Okay, they might have isolated from some organism, and they might have gone for some X-ray crystallography. Then only they might have got the structure. Yeah. So you have to prepare the target according to each and every, that's what I told, yes. So during this process, the crystallization process, or when they go for there must be some missing of chains, side chains, or missing of amino acids. So all these are preparatory steps. According to that particular tool, we have to prepare the target. Okay, the same thing ligands also. Again, you have to prepare the ligand, then you can go for the docking. And very importantly, evaluation of your results. Okay, so you people, you should have some understanding, then only you can have this evaluation, because when you click, the software will give you the results. Okay, within 5 minutes you will finish all the work, but how you are taking those results for your study, that interpretation is very, very essential, very much important. For that only, we used to tell you, study the concepts thoroughly. Then only you can properly integrate those results to your research. Okay? And this is the typical snapshot of the protein data bank. The moment you give any protein, you get the three-dimensional structure. Okay. Again, there are also some basic criteria. How to select the protein, how to select the proper PDB. If you get one protein, if you give one protein, it will give you n number of IDs, n number of 3D structures. Okay, so you have to be very clear. You can see here, as on, there are about 2,30,000 protein structures are available in this protein data bank. Okay. So these are the selection criteria. Okay, you have to be very clever in selection. The resolution should be less than 2 angstrom, and you have to know whether the complete structure is available or not. It is advisable you should go with always wild type protein. Okay, then the three-dimensional structures are available, the right conformation is available. So these are all some criteria like how you can go ahead with this PDB. Yeah. This is, I told, no, two things always you have to go. One is the score, another one is the two-dimensional interaction. You can see here, this is one particular ligand. So the ligand, it is having connection with the residues. This is the active site area. And the down, I have given one number. This is the binding energy calculation. Okay, minus 10.85. So one is the quantitative, another one is the qualitative analysis. And the second tool I will be taking quickly, ADMET tool. Okay, that is in silico ADMET prediction. Okay, we know very well that the drug molecule not only is expected to be efficacious, but to have unmet profile, which is normally expressed in therapeutic index. Okay, it should have a proper pharmacokinetic aspect. That means it should have desirable biopharmaceutical and pharmacokinetic properties. So for that also you have some ADMET prediction tools. Okay, yeah. This is some examples. For you have absorption. Okay, you can predict the oral absorption. You can predict the permeability. Likewise, you have distribution. You can predict the plasma protein binding and blood-brain barrier. Okay. Likewise, for each and every property, every aspect, it is connected to various biochemical or physicochemical properties. Then drug likeness, and this is a common rule for drug orally bioactive compounds. Lipinski rule of 5. And I have given some examples. Okay, so all these things are calculated through the various online molecular modeling tools. Okay, this is one particular tool, QuickProp. The QuickProp, it has given these properties, and each and every property you have some scale also. It should be within the limits. Okay. So CNS activity, it should be -2 to +2. Hydrogen bond donor, we should know very well, Lipinski rule of 5. Hydrogen bond donors, it should be less than 5. Hydrogen bond acceptors should be less than 10. Molecular weight should be less than 5. Molecular weight should be less than 500. Log P should be less than 5. All are multiples of 5, then only it is called, Lipinski has derived the rule of. Likewise, you have limits for each and every properties. These are all some tools, online tools. No need to pay anything. Okay? You can you will get the tutorials also. You can predict the properties. Okay, so I always choose this mole inspiration and Swiss ADME. Okay, especially for those who are doing final year projects, final year B.Pharm students or final year CAD elective subject students and all, you can use these tools very well and you can predict the properties. Swiss ADME, it will give you anything, bioavailability radar, okay, all these pharmacokinetic properties it will give you. This is the web page of Swiss ADME. Okay, so very simple. You can export the structure. You can import the structure from any publicly available database, or you can draw the structure using Marvin sketch. Okay, the the moment you click this button, the double arrow, okay, it will give you the smiles format of the particular drug. Then you should give run. Obviously, it will give you all the properties details. So this is one report, okay, where we have used that Swiss ADME tool and we have derived. You can see here, all the properties it will give you, starting from molecular formula, molecular weight, then what else, lipophilicity, water solubility. So all these details you can get it. The Swiss ADME tool example. The third very easily used tool is pharmacophore modeling. Okay, so pharmacophoric features, we should know very little basic. What are pharmacophore features? So these pharmacophore features are very essential characteristics which are needed for the interaction at three-dimensionally. Okay, so those are called as pharmacophoric features. So we should not confuse with functional groups, that is different. These pharmacophoric features, like very basic example I will tell you. So you have you have hydroxy group, you have NH2 group, amino group, you have mercapto group, SH group. Okay, all these three groups, what is the commonality? They all can donate hydrogen bonds. Is it? Are you all with me? Yeah. So those are called as what? Hydrogen bond donors. Though they are different, they have the unique characteristics to donate the hydrogen. So likewise, you have some 8 to 9 pharmacophoric features. Okay, so if you have those features, you can very well say, okay, the molecule can interact definitely with that particular target. Okay, so that is called as pharmacophoric modeling, and this is the IUPAC definition for pharmacophore. Okay. Yeah, as I told you, can see here. See, adrenaline, all these structures, they have some common characteristics. Okay, hydroxy group. Okay, this first drug has got 2 hydroxy groups and 1 NH group. All these 3 are hydrogen bond donors. Likewise, you have some unique characteristics, so those are called as pharmacophore. Okay, I will tell you the ligand-based pharmacophore modeling, it's a very good, you can excel in that particular area. Yeah, this is the very good example. Okay, you can see here, all these drugs, though they are different, they have a common pharmacophore. You can see 2 hydrogen bond donors, 4 hydrogen bond donors and 1 ring aromatic. Ring aromatic is a feature. Though it is, whether it is benzene, no problem. Whether it is naphthalene, whether it is pyridine, no matter. But it has got a ring structure. Okay, that is called as pharmacophore. These are the very important pharmacophoric features. HB, a hydrogen bond acceptor, HBD, hydrogen bond donors, hydrophobic aliphatic, hydrophobic aromatic, okay, negative charge, positive charge, negative ionizable, positive ionizable, at last ring aromatic. Definitely your compound will fall with any of these 2 to 3 pharmacophoric features. Okay, so now you can ask me, by this pharmacophore what I can do? You can go for pharmacophore based drug design. Okay, so the starting point is, all I should have the activity data. Minimum 20 compounds is enough. Minimum 20 compounds are enough. Okay, with that 20 compounds, there is a tool called as online tool, PharmaGist. PharmaGist, okay. So you can generate the pharmacophore. So that generated pharmacophore, you can give query to the large database. There are so many database, Zinc database, PEC database, okay. Like that, you can, first you can generate the pharmacophore, 4-point pharmacophore here. then it has been given as a query for a large data set of compounds. Okay, now what this query will do, it will pick up the like features. For example, from 1 lakh compounds, 1000 compounds will be filtered. Okay, so from that you can go ahead with either buy or synthesize whatever it is. So this is ligand based pharmacophore modeling. At last, virtual screening. I told one step, you have large data set of compounds. From that you can pick up, okay, according to your criteria or according to your need. Okay, so that is called as virtual screening. This is different types of virtual screening. Again, if you know the three-dimensional structure, you can go ahead with similarity search. Then pharmacophore mapping and machine learning. Structure based, you can go ahead with docking. Okay. So this is the types of virtual screening and molecular dynamics. So this is how the drug is behaving, because we know very well it's very much dynamic in nature with respect to time, how it is behaving in our physiological system. Usually the validation of your molecular docking, the structure based drug design, we go with this molecular dynamics. Yeah. So with these tools, I am going to explain a small case study, just 5 minutes, okay? How we have integrated the different tools, okay? Yeah. So the work is synthesis of HIF1 alpha hypoxia inducible factor 1 alpha mimetics for diabetic wound healing therapy for diabetic ulcer. We know very well that is a very common problem, okay, for the diabetes patients. We have integrated all the approaches in silico, in vitro and in vivo approach. This is the research gap, okay. So there is no direct drug for the treatment of this diabetic wound healing, okay. So these are all management of this disease, okay. You can see there is an ointment, okay. It is a recombinant platelet derived growth factor. Then you have some bioengineered products like dermograft, appligraft and all. What we try to do it, there is as such all management of these diabetic wound healing, okay, no direct drug. So what we thought, I told you from the beginning, identification of a target is very much important. So here we identified our target as hypoxia inducible factor, HIF1 alpha. Okay, so these are all some normal physiological process. Wound healing is a systemic and dynamic process involving all these epithelialization, angiogenesis and all. So what happens this hypoxia inducible factor? Yeah. So normally this hypoxia inducible factor, when the oxygen levels are low, okay, so normally what it does, it goes and binds with some protein which is called as Van Hippel Linde protein, and they form some complex and it will form some proteasome, it is degraded, all those things, okay. That and all we are not into it. Actually our idea is to inhibit this interaction. This HIF1 alpha and VHL protein interaction should be inhibited. So the HIF1 alpha will be very free, so that our design molecules, it will go and effectively bind with this HIF1 alpha. That's what our basic idea. And for that, here we have used this DHL inhibitor, this van hippale lindane protein inhibitor. Okay, so our idea is to find out some molecules which inhibit this interaction. So all these physiological process will happen. At last it will end up with increased diabetic wound healing. That's what our idea. Okay, this is our aim and objectives. Okay, how we are going to do this work. Yeah, so here I need your attention. So the second phase is in silico studies. So here we have used the visual screening, pharmacophore modeling and molecular docking, and we have predicted in silico level admit also. Okay, then afterwards we gone for the synthesis, characterization, in vitro, and finally in vivo studies. I am not going much in that part, synthesis, in vitro and in vivo or not. Yeah. So what tool I have used? Ligand based visual screening. So for that, collection of molecules, generation of pharmacophore and visual screening. Yeah. So these are all the existing molecules, the VHL inhibitors. Okay, so please bear with me for another 5 minutes. Okay, so these VHL inhibitors, I have derived some pharmacophore. Can you see the pharmacophore what we derived? It is a 3-point pharmacophore. We have derived. Okay, then with this derived pharmacophore, we went for So the pharmacophore model I have derived, that 3-point pharmacophore that has been given as a query in the large database. Here we have used zinc farmer database. Okay, so it has got lakhs together molecules. From that we captured some molecules which are exactly focusing or exactly matching to the pharmacophore what I have given. So all the web screenshots I have given here. And the selected molecules have again, it has gone for molecular docking studies with the particular VHL protein. Okay. Then after this molecular docking studies, again I have to recheck whether these are all safe or not. So we have gone for ADMET studies. Okay. And finally we have come to a conclusion that this analog can be taken up further. It can be synthesized. It is a pyrazole analog. So this what you can initially integrate 2 or 3 molecular modeling tools for your study. Of course, you can synthesize. You have a defined scheme, and always you have to make sure that whether your imagined or hypothetical molecules are novel. You always have to make sure. Okay, because around the globe, so many people will be doing the same research work. There is a tool, SciFinder. It will give you. Suppose if you are designing a new compound, whether this compound is new, obviously it won't be completely new. There will be some similarity. Matches will be there. You make sure that there will be no 100% match for your design compound with the existing molecule. So SciFinder, novelty, check we did. And I think these are the synthesized compounds. Physically then we have done. Okay. Then this characterization by various spectroscopical methods. Then finally biological evaluation. In vitro scratch wound assay we did. and in vivo streptozotocin induced diabetes model we have. And we concluded that this pyrazole compound will have or it may possess some diabetic wound healing activity. And these are the essential features, our own hypothesis it is. Okay? We propose the following structural features for diabetic wound healing therapy through HIF1 alpha mimetic pathway. It may have pyrazole nucleus, and very importantly, you can see the substitutions, it may play a major role. It may be phenyl group also. Okay, that's what we concluded. So, my take-home message is, initially, whenever you are planning for a simple research, you don't go directly to the wet lab. Okay? You can do some sort of molecular modeling tools. With available molecular modeling tools, you have some assumption or you have some prediction. With that, you can go ahead with your wet lab. Okay? So definitely. And I am telling you one more fact also, there won't be always a correlation. In silico and in vitro prediction, 100% there won't be correlation. You have again, there may be some pitfalls. Only thing is, we are having some prediction, having some assumption, you can go ahead for your wet lab work, for your further studies. The work has been published also in Royal Society of Chemistry. And so with this, I will be concluding my talk. So once again, I thank the management of Ultra College of Pharmacy for giving me the opportunity to share my experience to all the young minds. And my acknowledgement is my sincere pranams to His Holiness Swamiji, because I am coming from a Jagadguru Sri Shivaratrishwara Mat run academic institution, and my special thanks to all the leaders of JSS Ahar, especially Dr. B. Suresh sir, Professor Chinnaswamy sir, and my principal Dr. S.P. Dhanapal, and my own mentor Dr. Afzal sir. And always I am thankful to my own PG and PhD students, and I will be very much loving to be with my students. You can see I will be always with my students only rather than with my colleagues. Okay? And this is one book we have published, Computer Aided Drug Design, with my friend Dr. T. Prabha. It will give you all the basic insights. Okay? What all I have spoken, all these basic insights with the publishers. Of course, the publisher is also here, Dr. Jnana Prakash. So you can see all my profile. Okay, all my research works will be integrating to this computer-aided drug design. Of course, I make sure all these in silico tools will be validated by in vitro and in vivo techniques. Okay, so that's all from my side, and thank you for your patience hearing. Thank you.
Thank you, ma'am, for your informative message. It was really interesting and inspiring. By your docking studies, you opened our door of knowledge on this topic. Now this session is over. The delegates can raise your questions.
Sir.
Madam, it was very nice, eye-opening presentation on computer-aided drug design. I have some few questions. One basic question. Recently, I was working with one of the artificial intelligence-based company who is trying to put together a document on how to templates are created. Now, in the computer-aided drug design, whatever you have mentioned, the feeding is very important. So that becomes human intervention. What is actually you would like to tell to the student what to be fed first, right? Then computer gives outcome. Then what knowledge is required for the person to understand whatever is fed, whether computer aided design is giving the output which is as predicted as against available drug which is better predicted now. So what is that gap which you feel that should be linked, which is I think very important for the students over here, which will really help them to understand.
Yeah, it's a very good question, sir. Yeah, actually, they should have first knowledge about the pathophysiology of a particular area or particular disease. That's the first thing. And they should have basic knowledge about organic chemistry, sir. That is the second thing. And the third thing is my observation, my understanding for the past 15 years is many of the students, they are very good in doing all these usage of tools, all these things. But they are failing in interpreting the results. For example, one student, he is very good, he will within 2 or 3 days, he will derive so many hypothetical molecules, everything. But he fails like how it is applicable to the, what I can say, to the society or pharmaceutical research. For that, he should read lot of research papers where we are facing lot of problems there. So, they have to update, they have to read lot of papers related to that.
Yeah,
Yes.
that's what I want everyone to know that. Unless otherwise that is
Yeah,
known.
yeah.
Then there was a slide on 2D interaction.
Yes, sir.
You did mention on minus 10.3.
Yeah,
So can you explain what exactly
Yeah.
it means?
Sir, it is a score. It is a dog score. Usually, it is binding energy. That minus 10.8 kilocalories per mole. It is.
Okay.
So, the basic understanding is the lowest the binding energy, the stability will be more, or the affinity, not stability. I am not talking to the stability. The binding affinity will be more. The more negative values, the binding affinity will be more. And again, you rightly told, again it is a relative term. The student has to compare the value with the existing molecule. We call it as standards, positive standards. So with that, we should compare.
Yeah, so
Yeah.
in your case study, your prediction values are minus 7.
Yeah, it is
So-
one representation, sir, it is. Yeah.
Okay, so what is that actually required, the ADMA prediction parameter, whatever you
Yeah,
mentioned,
okay.
as against 7 for a topical wound healing, as against oral absorption? How these predicted values are interpreted?
Yeah. So again, these predicted values are standard values, sir. Like for example, if I want to design a compound with a very good, what I can say, very good triple B permeability. So it is a very basic observation. The molecule should have more lipophilicity. So the compounds which has nearing to 5, those molecules I can pick it up. Like that it is.
Log P value
Yeah,
and the Lipinski rule.
yeah, high log P value. It is a basic, like big general assumption that definitely it will have more, yeah.
Okay, and one more question, sorry. You did mention on the Swiss ADME drug
Yes,
design
sir. Yes,
database.
yes.
Whether this database works only when CAS number is available or if you know only the structure. See, I'm talking about an impurity. So we try to qualify on unknown impurity, where we need, if it is an unknown impurity, it doesn't have CAS number. So you'll have to generate a lot of toxicological data. Using this Swiss design, if I feed structure to
Yes,
it,
exactly,
it
sir.
will
Yeah.
give toxicological information.
Yeah, it will give you everything. It will give you, sir.
Without
Yeah.
CAS number also.
Yeah.
Without CAS.
There is two options, sir. One is you can retrieve from PubChem, the CAS number or smiles, it will be available there. You can take it. Otherwise, as you rightly told, you have the structure, you can very well. Because the Swiss ADME tool is inbuilt with Marvin sketch.
Okay.
So you can draw the structure using that and it will give you all the properties.
Thank you so much.
Yeah, thank you, sir.
Thank you, sir. It's time to honor our chief guest. As a token of appreciation, we would like to give a memento. I request our Vice Principal, Dr. P.S.S. Ramkumar, sir, to present the memento.
Hello. Dear students, I am really very proud that Jubi is here. She was my student in 1995 at Sankaling Bodhisari College of Pharmacy. She has developed her knowledge up to the research level and he is going to get many patents hereafter. You can see her name oftenly in the field of pharmaceutical chemistry. My heartiest congratulations to Jubi. So I am very proud that she was my student and that will give an enthusiastic admire for you all to be like Jubi. Congratulations, Jubi.
Thank you, sir, and thank you, ma'am.
It's a kind of reminder. I request all the students of each and every college to kindly fill their feedback form and our staffs will cover those forms before you leave.
Pharma science.